Antimicrobial use and resistance, will we ever understand it? I may answer that question by the end of the talk, we'll see. I'm going to ask your forgiveness right up front because a lot of the cases and the research that I'll be talking about today is from the beef world because I was at Colorado up until a couple months ago, but I'm very excited about learning more about what all you do in the swine world, and I think that you'll probably pretty easily see how some of these things that I talk about translate into your world and what you do on a daily basis. So I want to start by stating the obvious. And that's that antimicrobial resistance is complex. And I'm actually hoping to show how we might be able to leverage this complexity to our advantage and to the advantage of the animals that we work with, and also to the advantage of public health. But it is definitely a complex topic, and so we're going to dive a little bit into that complexity today. Because it's complex, it takes a whole lot of people to work on it and to work on it successfully. And so before I even start, I just want to acknowledge that what I talk about today has only been made possible by a lot of hard work by a lot of really great people who are all pretty passionate about answering this question. And that's how livestock production practices, and that includes practices around antimicrobial use, impacts antimicrobial resistance. And when I talk about resistance, I'm talking, of course, about human and public health, but also animal health and the health of the groups and populations of animals that we work with. And so I think one of the reasons antimicrobial resistance is so complex is because there's actually multiple layers of complexity. And I've listed two of them here, one being complexity of the systems that we work in, and the second being the complexity of the microbes themselves. And I'm going to go into more detail on each of these sort of layers of complexity because I think that it sort of tells us something about the system of resistance and how we can start to better understand it and also prevent it. So the first system I'm going to talk about is this first layer of complexity that I'm going to talk about is the systems level. And as an epidemiologist, and probably you all in this room, we can take a step back and think about where in our society do we typically use antibiotics. And so here I've just put some circles. These are some circles where I've listed where we use antibiotics in our society fairly regularly. And so there's some places that we're all aware of, human hospitals, farms, and then also in our companion animals, in industrial production of biocides, different things that we might not be aware of. And so if we're trying to understand how different use practices in these different spheres of our society impact resistance, it becomes extremely complex very quickly. So the details on this slide aren't important, but what these are all of the different pathways by which resistant bacteria and resistance genes can travel between all of these different compartments. And so if we as veterinarians and epidemiologists and researchers are interested in how what we're doing on farm in this orange bubble over here impacts something that's going on in human populations, we have to take into account all of these very complex and diverse pathways by which things can be transmitted and transferred. And so when I first was introduced to this topic of antimicrobial use and resistance, I came onto a study that was already ongoing, and it was really trying to design sort of the optimal study to look at associations between antimicrobial use and resistance, specifically in animal populations. So we weren't even trying to start on any of these long pathways that connect us to animals, but we wanted to really focus on a population of animals, and we wanted to overcome some of the challenges that we have in designing really robust epidemiological studies around this topic. And so some of the things that this study did is that we were able to accomplish some what we thought were fairly novel study design considerations. So we were able to conduct this study in real-world feedlots. So this was done in beef. We were able to utilize digital unique animal identification numbers for all the animals in the study, and those unique IDs were tagged to specific antimicrobial use and treatment records and also animal health records. So we had really robust digital data around all of these steers. And then we also did longitudinal sampling. So we went into these feedlots multiple times and sampled the same animals over and over. And of course we were able to do this using the chute, because this is beef production, and the chute had an RFID tag and it was all digitized. So it was really nice in that way. And that's honestly one of the challenges in doing these types of studies, is just keeping records of everything. And this was also a large sample size, which research becomes important because sometimes these use-resistance associations are fairly subtle. And so we had over 6,000 steers enrolled in this study, and we sampled them both individually for feces and then also deep nasopharyngeal swabs because we looked at Mannheimia. And I'm not going to talk about Mannheimia today, but I will talk about the fecal samples that we looked at. So for this study, we went into 4 feedlots, and again, we sampled 300 pens and 6,000 plus animals in those pens. We took the fecal samples, we isolated non-type specific E. coli, and then we tested those isolates of E. coli for resistance to a panel of 21 different antimicrobial drugs using 2 different methods. So DD is disc diffusion and BM is broth microdilution. And so we thought that this was a really robust way to look at associations between use and resistance because we were taking these fecal samples over time. So we could look at exposures and then what happened to the E. coli in terms of resistance. And so we developed a lot of models. We modeled these data in about 200 different ways. And in the end, what fell out was that parenteral tetracycline use did increase the odds of us isolating resistant E. coli from those fecal samples. Parenteral or injectable sulfonamide use did increase the odds of us isolating sulfazoxazole resistant E. coli. And interestingly, recent parenteral macrolide injections decreased the odds of isolating ampicillin resistant E. coli. So these were sort of the consistent associations that we found in this very large, multi-feedlot study. And I will point out that while these were statistically significant associations, they were fairly weak in terms of the magnitude of effect. So the odds ratios were somewhere between 1.1 and 2, so not huge odds ratios. The other thing I want to point out is that all of the significant exposures that we identified were recent. So the injection had to be given within 7 days of us taking the fecal sample, and only then did we find those associations. If we went further out in time, that increase in resistance disappeared. So it was sort of a short-term pressure on the E. coli. All of the significant exposures were parenteral, so we did have in-feed drug data for this study, but none of the in-feed drug exposures seemed to have any relation to resistance that we were seeing in the population. And the macrolide use was protective, so should we use more macrolides? Probably not. And so that really sort of caused us some pause, and really the biggest takeaway for me and for the group on this project was that the results were not at all consistent between the different models. So here we had designed this study that was very costly, very resource intensive, very difficult to undertake, especially if you're not using digital records. And we thought we had sort of solved all of those logistical challenges, yet the results that we were getting were sort of ephemeral and they weren't really robust. And so we sort of were a little bit disheartened. But at the same time, sort of serendipitously, these next generation sequencing technologies were coming out onto the market and being used more consistently. And so what we really thought about is, are we missing something? And are sort of the non-robustness of these results evidence that we're missing something big in this whole picture of antimicrobial use and resistance. And so that brings me to the second layer of complexity, which is about the microbes. And so I'm going to take a step back and give a little context about the complexity of the microbial population. So research for a long time, as you just saw in the study that I just presented, has been based on culture of specific bacteria. Sometimes that's pathogens, but oftentimes we talk about indicator bacterial species. So in the study I just presented, E. coli was sort of an indicator organism, and we say, well, if resistance is increasing in E. coli, we can assume that that's what's happening in the general population of bacteria. But really, because we can now access the entire microbial population, we know that a single fecal sample from a steer contains over 1,000 different bacterial species. And so this is a pie chart, and each little tiny wedge in there is an individual bacterial species that we identified in a single fecal sample taken from a feedlot steer. And so I think it's fair to question whether or not, E. coli, which is one of these little wedges, truly is representative of all of the resistance dynamics that are going on in all of these other bacteria within this population. And so that's really what we set out to try to understand, is if we look at all of the bacteria in a sample, do we get a different picture on what's going on, and can we better understand how our livestock management practices are impacting resistance? This is possible because of these next generation sequencing machines. This is just one model. And I'm going to quickly run you through what we term a metagenomic study. So a metagenomic study is where we take a sample, it can be anything, and we extract all of the DNA. And I want to point out that there's no PCR here, there's no culture, there's no enrichment for anything. We don't try to get rid of anything. It's just all there. So we extract all the DNA, we break it up into small fragments, we put little adapters on the end, and we then put them onto one of these sequencing machines. And the sequencing machine simply goes through and reads the sequence of DNA in all those short fragments. And it spits out basically a huge Word document of all of the A's, G's, C's, and T's that are within those fragments of DNA that you've put onto the sequencer. So when we take a sample, we see bovine DNA, we can see plant DNA, we can see, insect DNA, and we can see bacterial DNA. So we get everything, and so it generates certainly a whole lot of A's, G's, C's, and T's. So some people would term this big data. The computer scientists I work with say no, Google is big data, you're not big data. But regardless, one sample typically generates about a gigabase of data, and so a typical data set that has a fairly good sample size would generate terabytes worth of just A's, G's, C's, and T's in these short fragments. And so what we do is we take those short fragments and we look at the sequence and we try to match it to known resistance gene sequences that are available publicly in databases. And when we find a match, we say, okay, we found a match to resistance gene X, and we count the number of times that we find a match to that gene. And so by identifying and counting, we can then get a picture of what we term the resistome. So this is analogous to the microbiome. It's termed the resistome, and it's just the collection of all the resistance genes in their relative abundance within a given data set or within a given sample. So that's great. We have this new way to sort of count resistance genes, but how do we actually use it? And so this was our challenge when we were first getting into this area. And so I'm going to run you through a couple studies pretty quickly about how we can use the resistome to answer questions in maybe different ways, or to get a different perspective on what we're doing in livestock production to impact resistance. So the first study I want to talk about is a study that was done in 4 feedlots, 2 pens per feedlot, so 8 pens of cattle. And we went into these pens of cattle when they were placed in the feed yards, and we took soil, feces, and water from the pen. And then we let these cattle go through the feeding period. Nothing unusual was done in these pens. It was just standard feedlot operating procedures and management. And at the end, right before the cattle were shipped, we went back and resampled those same pens. The cattle were transported to the slaughter plant. We went and swabbed the insides of the trucks. The cattle then went into the holding pens outside of the slaughter plant where we took more samples. They then went through slaughter and fabrication, and at the end of that we actually swabbed the belts that were carrying the primal cuts from these cattle, and then also we took the trimmings, and that was a sample. So we had sort of this longitudinal snapshot of sort of these important phases of beef production, and we wanted to say how does the resistome, what does it look like? How does it change? Does it change? How big is it? How small is it? Does it differ as these cattle are being put through this system? And so again, we did the standard metagenomic approach with these samples, and in the end, when we got our sequence data, we had produced 4 billion reads. So reads are those little fragments of DNA, and 1.2 million of those reads aligned to something in the resistance gene database that we've developed. So, it's a small percentage of all of the DNA that we got that was actually a resistance gene. And those 1.2 million fragments of DNA, they matched to 319 different unique resistance genes in the database. The vast majority of those resistance genes were to tetracyclines and macrolides. So the real question is, okay, this is interesting. I can't tell you whether this is good, bad, normal, because we have nothing to compare it to. But this is what we got. And what we really wanted to see is how these genes are changing over the beef production system. So I'm going to focus on the results that we got for these in-plant samples, because I think they were interesting. So I can talk if you have questions afterwards about all the other samples, but I want to focus on this for now. Because interestingly, we had no matches to AMR genes in those post-slaughter samples. So the belts and the trimmings, we couldn't find any resistance genes in those. So at first we thought, okay, couple things could be going on. First of all, those samples were heavily contaminated with bovine DNA because we were basically sampling the meat, right? So maybe we have a sensitivity issue because we can't sequence through all the bovine DNA to find the resistance gene. So that's one hypothesis. The second hypothesis is, well, we know that from the multiple hurdle intervention technology that we assume that we're seeing big reductions in the bacterial population, right? These multiple hurdle interventions are targeted towards pathogens, but they're things that affect all bacteria, like high heat, lactic acid, things like that. So maybe the multiple hurdle interventions are not only reducing pathogen load, but they're also reducing the resistome as a whole. And we wanted to try to get a handle on which of these two hypotheses might be correct. And luckily we had the metagenomic data, so we could go back and look at the microbiome, so the bacteria and what was happening to the bacteria. So this is a busy slide, but what you see here on the x-axis is the log-fold change in abundance of different bacterial genera, comparing all the pre-slaughter and samples to all the post-slaughter samples. And then on the y-axis, we just have a measure of statistical significance, so anything higher up had a smaller p-value. And then each dot is a bacterial genus or a viral genus, or an archaeal genus. So this was our database. We had all of those. And so I've highlighted those genera that have at least they increased in abundance by at least 2 logs during the slaughter process. So for some reason, these bacteria did really well during slaughter. And interestingly, what you'll see is that a lot of them are thermophiles. So they are heat-loving bacteria, and they're hardy environmental bacteria or spore formers. And so interestingly, one hypothesis is that the multiple hurdle interventions are drastically decreasing most bacteria within the population. And you'll notice that most of the gray dots are to the left of the 0 line, so they indeed decreased in relative abundance during slaughter. But we see some of the bacteria thrive under those conditions, right? And those are the ones in yellow. And so this gives some credence to the theory that, the hypothesis that, the multiple hurdle interventions are indeed decreasing the vast majority of bacteria and the resistance genes that they're carrying. But we can't discount a lack of sensitivity to detect resistance genes as well. So this is one way I think it's interesting to use these types of data. And I think what it really told us was that metagenomics is really a worthwhile approach. We wouldn't have seen these patterns if we had looked at, just Salmonella or just E. coli. And so this was the first study that we had done using this, and I think what it really showed to us is we need to pursue this further. And so the next study that I want to talk about is a more narrow study, a more targeted study, looking at the impact of metaphylactic treatment, specifically telithromycin or Draxxin in feedlot cattle. And so in this study, we went into a feedlot when cattle were being processed, and we sorted the cattle into 2 pens. In one pen, all of the cattle received an injection of Draxxin at processing. In the other pen, none of the cattle received any injection of any drug, so that was our control pen. And at processing, we took individual fecal samples from these steers. And 11 days later, we went back. In the meantime, the cattle were treated the same in both pens, and we took fecal samples again. And of course, our biggest question was, does the resistome on day 11 look different between these 2 pens of animals? And if so, how does it look different? And so interestingly, there was no difference in the resistome between these 2 pens of cattle, either at day 11 or at day 0. So the resistomes looked very similar, and indeed the microbiome looked similar as well. What we did notice is that there was a huge shift in the resistome between day 0 and day 11 in both groups of cattle. And I mean, it was a very dramatic shift. And that was also reflected in the microbiome. So the bacterial composition was changing a lot. And so, this made us think, okay, if you really think about what we're doing with these cattle at this time, this is a huge transition period, right? They're coming into a new environment. They're being mixed with other cattle that they haven't been in contact with before. They've just been transported, so that's a stressful event. And their diet is being transitioned, right? They're heating it up. So we think that those livestock management factors probably play a much larger role in shifting the microbial population and those resistance genes than even something like metaphylactic treatment where every animal is being exposed. And so we think that this is a really important finding because it shows us that the larger context is important when we're looking at the microbial population and when we're really interested in understanding how is the population of bacteria responding in terms of resistance. And so the key takeaway for me on this study is that it's not just about antimicrobial use. And this is something that I really hope sort of I can drive home in this talk, which is that in my opinion, we need to be taking a much broader perspective when we run these studies in terms of when are we using antimicrobial use and how does it fit within the larger context of what we're doing with the animals, right? So that study has been published, and so you can read about that in more detail, but I'm also happy to answer questions. The third thing I want to quickly cover is something else that we can do with metagenomic data and with resistome data, and that's that we can start looking at so-called resistance fingerprints. So this is again, sort of a busy figure, but across the top I've put the TET-L gene, and these figures are taken from fecal samples that we collected in a pork system and a poultry system. And so across the sort of x-axis you have the TET-L gene. It's about 1,900 base pairs long. And in these little windows, I'm going to blow one up here, each of those rows going across is one of those fragments of DNA that we sequenced on the sequencer. And the A's, G's, C's, and T's are the sequence of that fragment. And where the fragment of DNA from our sample matches the reference TET-L, we have a gray box. But where there's a change, we have a red box. So that's a SNP, or a variant, right? And so what you see is that for the pork samples, we have these longitudinal red lines very consistently in these certain areas along the TET-L gene. When you look at the poultry, you don't see that sort of any of those red lines. So I'm not saying whether or not these changes, these variants, these SNPs are good or bad. We don't know. But what I am saying is that for this study, and for this specific set of samples, we could differentiate the samples that came from pork versus those that came from poultry based on this pattern. And this should probably be ringing some bells in terms of like PFG and MLST typing and then now whole genome sequencing. We can do it at the resistance gene level as well. And when you think this is just one gene, this is TET-L, but we found 319 resistance genes in that study that I presented first. You can do this for all 319 genes. Do we have now a very powerful way to start tracking resistance across this whole system, right? And so this is something that we're working on right now to understand, how discriminatory are these fingerprints? How unique are they to different environments? We have no idea. Maybe the pork fingerprint is common to beef cattle. I only showed poultry. I don't know. So we have a lot of work to do as epidemiologists and scientists and veterinarians to understand really how to interpret these data. And that's really what I want to make sure I emphasize heavily is that it's, in my opinion, still early days for these technologies, especially in livestock production settings. And I think that we haven't answered the so what question. So we find a specific resistome profile and it shifts. Is that good or bad? I cannot tell you right now what a bad resistome looks like and what a good resistome looks like. And I think that that's critical information to assessing risk and to assessing how what we're doing as producers and veterinarians is impacting the microbial population and resistance in the environment, in our own herds, and in people. And so with that, antimicrobial use and resistance, will we ever understand it? I actually think that we now actually have the tools to understand some complexity, at least enough. And by enough, I mean to actually produce some robust evidence-based understanding of how the microbial population is responding to what we're doing at a systems level. And so with that, I have a lot of people to thank. I've listed some of them here.